Hello friends, welcome to entranceadvisor.com. Today's topic is the molecular basis of inheritance class 12 biology, that is the chapter 6. So let's start the video. Before starting the video, if you are new to this channel, please hit the bell icon and do subscribe to our channel. So what is DNA? Before talking about this, you should know something. The extracted acid material from the cell nuclei is called the nuclein, named by Meister, that proved to contain an unusually large amount of phosphorus and contains no sulfur. In the latter years, the nuclein was named as nucleic acid that was found to associate with various proteins. The nucleic acid combined with the protein is known as the nucleoprotein, which is the main chemical composition of the eukaryotic chromosome. The nucleoprotein is found to be formed with the DNA and histone proteins, although some amount of RNA and non-histone proteins are also present in the chromosome. So DNA or the RNA are the two types of nucleic acid found in the living organisms. And the DNA acts as the genetic material in most of the organisms, and RNA also acts as a genetic material in some organisms, in some viruses, and acts as a messenger. It functions as an adapter, structural, and in some cases as a catalytic molecule. The DNA, it is a long polymer of deoxyribonucleotides. A pair of nucleotide is known as the base pairs. The length of the DNA is usually defined as the number of nucleotides present in it. The Escherichia coli have 4.6 into 10 to the power 6 base pairs, and haploid content of human DNA is 3.3 into 10 to the power 9 base pairs. So let's talk about the structure of the polynucleotide chain. So as you can see in this chart, it's given the nucleotide is composed of nitrogenous bases, pentose sugar, and phosphate group. Nitrogenous bases is further divided into purines, that is adenine and guanine, and also the pyrimidines, cytosine, uracil, and thymine. The pentose sugar is divided of two types, ribose and deoxyribose. The nucleotide has three components: a nitrogenous base, a pentose sugar, and a phosphate group. There are two types of nitrogenous bases, purines and pyrimidines. Cytosine is common for both DNA and RNA, and thymine is present in DNA. Uracil is present in RNA at the place of thymine. And the nitrogenous base is linked to the pentose sugar with N-glycosidic linkage to form a nucleoside, when the phosphate group is linked 5' OH of a nucleoside through the phosphoester linkage, and the nucleotide is formed. The nucleotides are linked through 3' to 5' phosphodiester linkage to form a dinucleotide. This is a polynucleotide chain. As you can see, the more nucleotide joins together to form the polynucleotide, and in the RNA, the nucleotide residue at an additional OH group present at the 2' position in the ribose, and uracil is found at the place of the thymine. So the nucleic acid is the organic acid which is found predominantly within the nucleus and takes part in the formation of the chromosome, is known as the nucleic acid. The composition of the nucleic acid, that is, it is made up of nucleotides. A nucleotide is a small urinary structure of the nucleic acid. Each nucleotide is composed of a nitrogen base, pentose sugar, and the phosphoric acid. A nucleotide without a phosphoric acid is known as the nucleoside. This means the nitrogen base and the pentose sugar together without the phosphoric acid is known as the nucleoside, and N base plus pentose sugar equals to nucleoside. N plus pentose sugar plus phosphoric acid is a nucleotide. The structure of the different components of a nucleotide is as follows. I am talking about this in a brief, very quickly. The nitrogenous components present in the nucleotide is known as the nitrogen base. There are two types of nitrogenous bases, such as pyrimidine and purine. The pyrimidine, it is a single ring molecule of the low molecular weight in a DNA molecule. The pyrimidine bases are cytosine and the thymine. And in the RNA, the molecule pyrimidine bases are cytosine and uracil. The purine, this is the two-ring molecular structure of higher molecular weight than the pyrimidine in DNA or in the RNA. The pyrimidine molecules are adenine and guanine. Okay. And the pentose sugar, the sugar molecule with the 5 carbon atoms, is known as the pentose sugar. Its chemical formula is C5H10O5 and it is also known as the ribose sugar. The nucleic acid which contains the ribose sugar is known as the ribonucleic acid or the RNA. In the ribose sugar, if one oxygen atom is deleted from OH group present in the second carbon atom, the sugar is now known as the deoxyribose sugar, and the nucleic acid that contains the deoxyribose sugar is known as the deoxyribonucleic acid or the DNA. And at last, the phosphoric acid H3PO4. This is the part of the nucleic acid and is present in the DNA and in the RNA in the same form. So here is the differences between the RNA and the DNA. DNA is double-stranded, RNA is single-stranded, structured into 2 chromosomes, and it is ribose, it is deoxyribose. It uses thymine, it uses uracil. It's modified 5' and 3' polyatyl. It both uses A, C, and G, has sugar plus phosphate backbone in common, stores genetic information read by ribosomes to form the proteins, and can self-replicate, can have multiple structures like this, can last for a long time. It's read by polymerases and reads by ribosomes and stores information about the protein structure, as I said, and does not usually last for a long time. It reads 3' to 5' and it's formed in 5' to 3'. So here you can refer to this image also. Its acid name is deoxyribonucleic acid and it's ribonucleic acid. Its stability is very stable, long life. It's less stable and it's present in nucleus, mitochondria and plastids. It's only in the limited to the nucleus and the cytosols only. And the function, the static, the digital genetic data storage, and it is dynamic. It's various forms and its various functions. DNA polymerases, it's long chain nucleotide made up of and 2 complementary strands A, B, C form helix, and RNA polymerase is of short nucleotide chain, only one strand, only A chain is formed, and backbone it's formed of deoxyribose and ribose as I said, and nucleobases are thymine, cytosine, adenine, guanine, and its uracil is present in case of thymine. Base pairing is A to G and C to G, and here it's A to U. EM radiation somewhat UV sensitive and relatively UV resistant. Apart from this, there are many differences too. You may refer to any of good books which are available. Okay, let's move on. So let's talk about the double helix model for the structure of DNA. James Watson and Francis Crick discovered this. Based on the X-ray diffraction data produced by the Winklin and Rosalind, proposed this model of DNA. The salient feature of this model are the DNA is made up of 2 polynucleotide chains in which the backbone is made up of sugar phosphate and bases projected inside it. So it is the double-stranded nucleic acid molecules that contain the deoxyribose sugar and in which the genetic information is encoded as a sequence of purine A, G and pyrimidine C and T bases attached pairwise by hydrogen bonds is known as the deoxyribonucleic acid or the DNA. So DNA is mainly present in the nucleus, as I said, in the prokaryotic and eukaryotic cells. In eukaryotic cells, it remains combined with a protein and to form the nucleoprotein. DNA is generally present in the chromosome. It is also found in the mitochondria and chloroplast in less quantity, and RNA is present in the cytoplasm, nucleus, and the chromosomes. The chemistry of DNA, all living organisms contain the nucleic acid in the form of the deoxyribonucleic acid, and DNA consists of repeating structural unit called the monomers or the nucleotides. Each nucleotide consists of pentose sugar, phosphate, and the nitrogen base. So, Watson and Crick proposed in 1953 this model. Okay, the DNA is composed of 2 antiparallel polynucleotide chains or strands that form a double helix around the central axis. The 2 strands are intertwined in a right-handed direction. So 2 chains. Here it is. 2 chains have antiparallel polarity, one from 5' end and with a 3' to 5' end. The bases in 2 strands are paired through hydrogen bonds, and adenine and thymine forms a double hydrogen bond, and the guanine and the cytosine forms a triple hydrogen bonds. 2 chains are coiled in the right-handed fashion. The pitch of the helix is 3.4 nanometer and roughly 10 base pair in each turn. The plane of one base pair stacks over the other in double helix to convert the stability. You can see the pictures of Rosalind Franklin, Watson and Crick, and in the basis of this extra diffraction which I showed you earlier, the picture was given in the first slide, and this is the structure they formed. So the helix makes one complete turn in every 34 Armstrong and has a diameter of 20 Armstrong. You can see this clearly. Here it is the actual original picture of them showing the structure, and you refer to this what I am showing now. So the difference is the distance between the two adjacent bases in a helix is about 3.4 Armstrong. You can see. Okay, here it is mentioned, and that means there are about 10 bases present in each helix for a complete turn of DNA. And there is one major group and then one minor group. This is major group and this is minor group in each complete turn of the DNA. You can see each complete turn there is. And the main axis of each helix is formed by the sugar phosphate backbone, and the nitrogen bases are stacked inside the helix in the perpendicular to its axis. And the two strands are held together by the hydrogen bonds. You can see these are very strong and established between the pairs of the purine and the pyrimidine bases. And the base pairs that occurs are adenine and thymine held together by the two hydrogen bonds. A is with the T and double bonded, and guanine with the cytosine held together by the three hydrogen bonds, guanine and cytosine. Thus the latter pair is more stable because there are three hydrogen bonds. And the axial sequence of the bases along one polynucleotide chain may vary considerably, but on the other chain the sequence must be complementary, as in the following example. This is not necessary, and because of this property, given an order of the bases of one chain, the other chain is exactly complementary to each other. And 3' to 5', this is the direction, the polarity of the two helices are opposite to each other, and the helix are therefore known as antiparallel. Okay, let's move on. Now the central dogma, what is that? The Francis Crick proposed a central dogma in the molecular biology too, which states that the genetic information flows from the DNA to RNA to protein. Okay, the DNA, its replication occurs, then DNA forms, and on transcription the mRNA forms, and on translation the protein is formed. This very simple strategy or the process is known as the central dogma. I'll be discussing in detail about this in the next videos. So let's talk about the packaging of the DNA helix. How they are packaged. In prokaryotes, well-defined nucleus is absent and negatively charged DNA is combined with some positively charged molecules called the nucleoids. In eukaryotes, the histones positively charged molecule organize to form the 8 molecules you need called the histone or the octamer. The negatively charged is wrapped around the histone octamer to form the nucleosome, and the histone is rich in basic amino acid residues, lysines and arginines. Both the amino acids residues carry the positive charges in their side chains, and the side single nucleosome contains about 200 base pairs. The chromatin is repeating unit of nucleosome. In nucleus, some region of the chromatin are loosely packed and strains light, and they are referred to as euchromatin, and the chromatin that is more densely packed and stains dark are called heterochromatin. The euchromatin is transcriptionally active chromatin, while the heterochromatin is inactive. Okay. Here are multiple pictures in order to understand you. This is the structure of a nucleosome. As you can clearly see, this is the DNA, and this is the histone octamer. It's composed of 8 proteins, different H2A, H2B, H2 and H3, and it's histone protein. You can see they are wrapped. The DNA in between them is known as linker DNA, and it's very hierarchical nucleus to chromosome to centromere to telomere, then nucleosome, histone, nucleotides. It's given hierarchical order. It's given in, and its DNA, it's 8 histone core. Only this 4 proteins, histone proteins, are repeated in the lower portion also. So this is very important, as you need to practice it a lot. many questions will come from this related to this new nucleosome model of chromosomes. If you want me a detailed video how the solenoid model works and how the nucleosome structure is set one another, that nanometer level of hierarchical order, I'll make a separate video for that. Now, the search for genetic material, the transforming principle, the Frederick Griffith in 1928 conducted an experiment on bacteria, Streptococcus pneumonia, bacterium responsible for the causing pneumonia disease. There are two types of strain called these bacteria. Some produce smooth, tiny colonies, S, and others produce rough colonies. The mice infected with the S strain virulent die from pneumonia infection, but mice infected with the R strain not develop the pneumonia. Make it a clear understanding by doing this, because this experiment will be important for your rest of your life, and this is very important. And S strain, when injected to the mice dies. When R strain injected into the mice is alive. When S strain, that is a smooth with heat killed, injected into the mice lives, and S strain, which is heat killed, along with the R strain, which is living, injected to the mice dies. So Griffith concluded that the R strain bacteria that somehow transformed by the heat killed A strain bacteria, some transforming principles transformed from A strain to R strain and enabled the R strain to synthesize a smooth polysaccharide coat and become virulent. This must be due to the transfer of the genetic material. Let's discuss this in very detail. So Frederick Griffith, first of all, I must say the definition of a genetic material, many of you don't know. The complex organic macromolecule present in the cell that carries genes or the message for the biological characteristics from one generation to the next is known as the genetic material. And the experimental proof of DNA as a genetic material was initiated by Frederick Griffith in 1928, first performed experiment on the bacterial transformation. So, the definition of bacterial transformation, what is that? The process by means of which the genetic modification of a bacterium occurs by addition of DNA in the bacterial culture by entering this foreign DNA into the bacterial cell is known as bacterial transformation. With the help of transformation experiment in mice, Griffith showed that one strain of bacterium is transformed into another strain. The Griffith performed the experiments on mice with a pneumonia disease causing what? You know this, I have just said, causing the pneumococcus bacterium known as the Streptococcus pneumonia. Normally, the bacterium cell is covered by the cell wall made of polysaccharide. Such bacteria from the smooth colony and are known as the smooth type or S type, and this bacteria can cause pneumonia disease and are harmful or virulent. As a result of mutation, the bacteria cannot produce cell wall. Such bacterial colony appears rough, and the bacteria are known as rough type or the R type, which cannot cause disease and are known as avirulent type. Make it a very clear, okay? And when bacteria are exposed to high temperature, they lose the capacity to divide, and such bacteria are called the heat kill bacteria. A particular bacteria may be found in different strains, that is 1, 2, 3, 4, etc., and that are genetically different. And what is the experiment? Mainly, the experiments performed by the Griffith were in vivo experiments. It's inside the lab, and as the experiments were performed within the body of the mice, okay, and the experiment are follows. As follows: the smooth strain is virulent, okay, that is very smooth and is virulent, is very harmful, and rough strain is not harmful, not causing the disease. And the heat kills smooth strain, as I said, and at last taken the rough strain and the heat killed along with the smooth strain. So mice was not killed when the virulent or the rough strain is injected in this case, and the mice dies when the smooth strain is injected was killed due to the 3S virulent bacteria were recovered from the body of the mice. These were recovered from the mice, and the experiment 3 in its causes, the mice was not killed when and no bacteria were recovered from the body of the mice when injected with heat killed. So, and the mouse dies when the rab strain along with the heat kill was injected. So, this is very clear that heat kill 3S bacteria has no power of division, and in this bacteria could not multiply and cannot cause the disease. And at last, the R3 bacteria with 3S heat kill bacteria injected into the mice was killed, and the 3S virulent bacteria were recovered from the body of the mice. And this 2R or the 3S or heat kill bacteria could not cause any disease when they are administered separately into the body of the mice, but a mixture of these two types of the bacteria when injected can cause the disease in the mice. It appeared that the rough bacteria somehow was transformed to smooth type of bacteria, and the Griffith proposed this phenomenon as the bacterial transformation. According to the Griffith, in this experiment, the bacteria was transformed into the virulent type. The Griffith called this phenomenon as the bacterial transformation, and the Griffith suggested that an active principle or the active substance was released from 3S heat killed and are transformed to R-type bacteria into the 3S type. Okay, Griffith was unable to find out the chemical structure of the active principle. So, after 16 years of Griffith's experiment, in 1944, three scientists, Averitt, MacLeod, and McCarthy, experimentally proved that transforming principle of Griffith was nothing but DNA, and thus they proved that DNA is a genetic material. So, experiment of the Oswald, Averitt, Colin, MacLeod, and McCarthy worked out to determine the biochemical nature of transforming the principle of the Griffith. So they purified the biochemicals and from the heat-killed S cells to see which one could transform the live R cells into the S cells. They discovered that the DNA along from the S bacteria causing the R bacteria to become transformed. So they concluded the DNA as a genetic material. The experiment performed by Averitt and his co-workers are known as the in vitro experiment, as the experiment was performed in a test tube or the dish using the artificial culture medium and not within the body of the organism. The experiment was just like that only. You can see the live two bacteria, the R2R, that is the rough strain bacterial cell grown in a culture medium or a petri plate or a test tube. The R colonies were formed, and in the separate case, the DNA formed is heat kill type with the culture in the petri dish. No colony of the bacteria was formed, and then inference was that the DNA of the 3S killed bacteria had no power of reproduction. So the DNA extracted from the heat killed cells did not respond or reproduce to form a colony of the cells. At last, when 2R or the rough type or the DNA of the 3S heat killed serum cells were cultured in a petri dish, incubated in a petri dish, or the transformation occurred, 3S colonies were found in the culture. So inference was that this experiment of Averitt is very similar to the fourth experiment performed by the Griffith, and Griffith used heat-killed 3S bacteria, but Averitt used the DNA from the heat-killed 3S bacteria. And Griffith performed the experiment on mice, but Averitt performed the experiment on the culture medium taken on a petri dish. The experiment results are similar in both the experiments, and therefore the active principle of the 3S heat-killed bacteria is nothing but the DNA. So in this slide, I'm going to talk about the experimental proof that the DNA is the genetic material was formed by the viral transduction method. So what is the viral transduction or transduction? The process by means of which the genetic material of one bacterium is transferred to another bacterium with the help of a phage virus is known as the transduction. So Alfred Hersey and Martha Chase in 1952 worked with the virus that infect the bacteria called the bacteriophages. In one preparation, the protein was a part of a radioactive, and in the other, the nucleic acid DNA was made radioactive. These two phage preparations were allowed to infect the culture of the E. coli soon after the infection before lysis of the cells. The E. coli cells were gently agitated in a blender to loosen the adhering phage particles, and the culture was centrifuged. The heavier infected bacterial cells are plated to bottom, and the lighter viral particles were present in the supernatant. It was found that when bacteriophage containing the radioactive DNA was used to infect E. coli, the pellet contained the radioactivity. If the bacteriophage containing the radioactive protein coat was used to infect the E. coli, the supernatant contained most of the radioactivity. His experiment shows that the protein does not enter the bacterial cell and only DNA is the genetic material. Here is the perfect diagram for this. You may refer to this. So, in the autotropic strains of the uh, virus that can destroy the bacteria is known as the bacteriophage. The body of the bacteriophage consists of the two components: other outer capsule, where it is the outer capsule, and the inner DNA. Okay, the inside the DNA is present. The bacteriophage known as T2 can infect the E. coli bacteria and the numerous viruses are formed. It has been found that in the capsule the phage virus sulfur is present and in the DNA phosphorus is present specifically. Utilizing this property of the phage virus, Harsey and Chaz labeled the capsule with the 35S radioactive sulfur and with the DNA with the 32P radioactive phosphorus. Okay, they labeled it. In the first experiment of the Harsey mixed with the 32P level with the T2 bacteriophage and E. coli bacteria for the 10 minutes for infection of T2 phage to the bacterial cell. The DNA of the phage entered the bacterial cell, but the protein coat would remain outside the bacterial cell wall. These bacterial cells were agitated with the help of a wiring blender to release the phage capsule from the bacterial cell wall. The bacterial culture then centrifuged and obtained pellet and supernatant were examined separately for the presence of radioactivity in the content. In the second experiment, the Harsey and Chase performed the similar experiment using the 35S leveled bacteriophage. The two sets of experiment are illustrated as I said. So if you want more explanation, then I'm explaining. And in the first experiment, the 32 labeled bacteriophage was used and the radioactivity in the pellet is found is very high and radioactivity in the supernatant is found is very low. The explanation according to the Harsey and Chase, the 32P labeled DNA of 32 phage enters the bacterial cell, but the protein coat remains outside, as you can see. As the pellet contained the bacterial cell, it is found almost without radioactive leveling, and trace amount of radioactive leveling is found in the supernatant due to the presence of some labeled phages that fail to enter the bacterial cell. And in the second experiment, where 35S level T2 phage was used to infect the bacteria, the supernatant was found to be highly radioactive and the pellet was with a low radioactivity. Explanation is that according to the Harsey and Chase, the 35S level protein capsules of the T2 phage do not enter the bacterial cell, and these capsules are present in the supernatant. This causes the high radioactivity in the supernatant. On the other hand, the sum of the radioactive capsule of the phage virus still adheres to the bacterial cell, and this causes trace amount of radioactivity in the pellet. Okay, I think you got it, that the DNA of the phage virus is responsible for carrying the characteristics to the next generations only, so they prove the DNA is the genetic material. So what are the properties of the genetic material? It should be able to generate its replica, that is the replication, very important, and it should be chemically and structurally be stable. It should provide the scope for slow changes, the mutation that are required for evolution. It should be able to express itself in form of the Mendelian characters. DNA is chemically less reactive but structurally more stable as compared to DNA. So DNA is better genetic material. So RNA was the first genetic material and used as a genetic material as well as catalyst and most reactive, so less stable. Therefore, the DNA has evolved from the RNA only. That is known as the RNA world hypothesis. Okay. Hope you learned something from this video. I explained more deeply than you thought. So thanks for watching. If you like this video, please comment, share and subscribe. Until then, take care and goodbye.